Chapter 13, Topics 13.4 and 13.5, Stages 3 and 4, T-cell and B-cell response. In this topic, I want you to be able to list the three major types of cells that T-cells will differentiate into after stimulation, describe the main functions of these three types of T-cells, explain how cytotoxic T-cells, or TC cells, kill other cells, diagram the steps in the B-cell response, make a detailed drawing of an antibody molecule, explain the various end results of antibody binding to an antigen, list the 5 types of antibodies and important facts about each, and describe the memory response. So in this topic, we're going to cover cell-mediated immunity, or CMI, activation of T cells and their differentiation into subsets, T cell subsets, B cell clonal expansion and antibody production, antibody structure and characteristics, antibody functions, types of antibodies, and primary and secondary responses. Let's be honest, cell-mediated immunity, or TMI, is complex. There are several different subsets of T cells, and they all end up doing something different. They arise based on who talks to whom, and there is an entire branch of science devoted to figuring all this out. I don't want you to know all of that for this class. What I do want you to know are the end results of stimulation. That would be the mobilization of B cells, phagocytes, and other T cells. Also, some T cells act to suppress the immune response. You don't want the immune response to run rampant once the threat is over. So with that as a framework, let's look at the activation of T cells. The phagocyte, or antigen-presenting cells, finds the T cell that matches the antigen it is carrying around. This activates the T cell. The T cell divides. The clones of the original cell can become any one of these subsets. Memory T cells sit back and wait for the next time the phagocytes come around with that antigen. There are more of them, so the second, third, etc. immune response will happen more quickly than the initial immune response, because the T cell is already activated. Some become T helper 2 cells, or Th2 cells. These helper cells go and activate B cells, which become memory cells, plasma B cells, and B cells that regulate other B cells. All of this stimulates the humoral response, which we will talk about in more detail later. Th1 cells stimulate macrophages to go out and eat more and eat faster. It's like giving an antacid to a full phagocyte. Th17 cells are involved in inflammation. T regulatory cells, or TR cells, are repressor cells, which dampen the immune system once the danger is past. They also tell the other immune cells to cool it if a non-pathogenic antigen is contacted. In other words, they prevent allergies. Some T cells have CD8 receptors. They become cytotoxic T cells. These cells undergo clonal expansion, and some become memory cells. Others go out and look for cells with their antigen. If they shake hands with the cell that is displaying its antigen on its MHC1, the cytotoxic cell releases porphyrins and granzymes. The porphyrins make holes in the membrane so the granzymes can get in and destroy the infected cell. This protects against specific viruses that you've encountered before, and other intracellular parasites that, once again, you've been exposed to before. There have been recent advances in teaching cytotoxic T cells to take out specific cancer cells as well. Here is a nice summary table on the different types of T cells. These are the only ones you need to worry about. So, on to the B cells. So, a T helper 2 cell comes and shakes hands with the B cell, activating the B cell. They also do some chemical chit-chat between each other. Some, after they divide, become memory B cells, which function the same as memory T cells, waiting and ready to divide the next time the antigen shows up. Most of the clones become plasma cells. Plasma cells produce tons of antibodies. In fact, they produce so many antibodies that they wear themselves out in 3 days. This is a good reason to have memory cells around so that they can restock the plasma cell population. The antibodies go out and do their thing. So, what is that thing? Why, to bind antigen. Here is a reminder of the structure of an antibody. So here's the Fc portion, and here is the antigen binding site. The antigen binding site has a variable region that is very specific for just one type of antigen. Actually, it's very specific for just one type of epitope. So once the antigen is bound, what happens? Antibodies can coat pathogens, inhibiting their normal function and reproduction. That means neutralization of viruses, which amounts to the same thing. When antibodies bind toxin, they are also neutralized, as their active sites, if they're an enzyme, are covered up or plugged up. Antibody-antigen complexes are more easily phagocytized, which is opsonization. Antibodies can grab on to more than one antigen. If the antigens are on other pathogen, it clumps them or agglutinates them. This makes it so the phagocyte can swallow down more than one pathogen at a time, leading to faster clearing. Complement will bind to the Fc portion of the antibody, leading to the classical complement activation pathway. Antibodies come in 5 types and they have different functions. Let's start here with IgM, because that's what your body starts with. Anytime you respond to an antigen for the first time, you will see more IgM in the blood than other types of antibodies. IgM is a pentamer, or it's 5 antibodies that are combined. They're really good at agglutination, but they're expensive to make. In subsequent immune responses, less IgM is produced. IgG is produced in the first immune response, but not as much as the IgM. Its levels really take off in subsequent immune responses. It is the most common antibody found in the blood. It is a monomer, meaning there's only one antibody, and making it fairly inexpensive to make. It is also able to cross the human placenta, providing protection for fetuses. IgA is a dimer, and it is found in secretions such as mucus and breast milk. It helps protect you from respiratory and gastric pathogens, and it helps protect little babies. IgE is found on the surface of mast cells and basophils. They are involved in triggering the release of histamine. As a part of that, it is implicated in allergies. It is also found in higher quantities in Helminth infections. You are probably wondering why I made such a big deal about how much is produced, when, and why. We can use this information to diagnose illnesses. Say, for example, I have a sore throat that won't go away and I'm tired all the time, even though all I do is sleep. My general physician thinks that I may have mononucleosis. But, and here is the important question, is this an reactivation, or is this the first time I'm getting it? Why, you ask, doesn't matter. Well, if I get mono as an adult or teenager, I'm going to be sick for longer and I'm more likely to have later reactivations. My GP wants to put me on antivirals if that's the primary infection. If it's a reactivation from mono I picked up as a preteen, yes, it does happen. Think about kids in elementary school sharing sodas or sucking on the drinking fountain. If it is a reactivation, my prognosis is much better. I won't be as sick for as long, and antivirals aren't indicated unless I've got something else going on. So, how do we tell? Why? By looking at the different levels of the different types of antibodies. If it is a primary response, I'm going to have lower amounts or titers of antibody, total antibody, than in a secondary response. And most of that is going to be, IgM. If it's a reactivation or a secondary response, I'm going to have more IgG than IgM. And I'm going to be, like I said before, having a higher titer of antibody. So hopefully you will now be able to list the 3 major types of cells that T cells will differentiate into after stimulation, describe the main functions of these 3 types of T cells, explain how TC cells kill other cells, diagram the steps in the B cell response, make a detailed drawing of an antibody molecule, Explain the various end results of antibody binding to an antigen. List the 5 types of antibodies and important facts about each, and describe the memory response.